with increased risk of breast cancer. So this is quite alarming. Even though this is just a pilot study, it's already alarming to us. So we wanted to study this further. I think this is very important to look into. In addition, these long chain PFAS seems to be correlated with age as well. This is not surprising because PFAS are high carbon and they are hydrophobic, and so they can stick to fat tissues and they can stay longer in the body, and it's a bit difficult to get rid of. So what we see is that with increasing age, you will also have high levels of these PFAS, and it seems like the longer chains also tend to stick longer in the in women. We didn't see this in other EDCs, this accumulation or this correlation with age. So this seems to be specific with PFAS, at least for the samples that we analyzed. Another thing that's alarming is trying to find where these PFAS are. So we did a simple PCA analysis and found that this triangle that you see, this dark triangle, these are women who are in region 4A, the Calabarzon area. And then you can also see these arrows, and these arrows are pointing towards these high levels of PFAS, sorry, the longer chain PFAS. And you can see that several individuals who are living in Calabarzon area seem to have, are the ones who have high levels of these long chain PFAS. So that is quite alarming because it tells us that maybe there is a certain group of individuals that have higher exposure to this particular chemical, and maybe they are also at risk of getting breast cancer. Of course, this is very early. This is a pilot study. We only analyzed 150 samples, 100 is this 100 something individuals, and so we're still trying to. So we need more data to try and see if this is true. Well, the source, of course, we still need to determine where this is coming from. You know, we know that there might be in individuals in region 4, but we don't really know where it's coming from. What we can, you know, what we know about the region 4 is there's a lot of industrial, there's an industrial park there. But of course, it's, you know, it's difficult to pinpoint on that. And plus, we don't want to step on other people's, you know, it's quite a controversy. It's very difficult to challenge industry. But of course, we need to try and figure out where these chemicals are coming from. It could be in the industry, it could be in the water. You know, some people are also thinking about the lakes that are there in that area. And hence, it's very important for us to collaborate with environmental chemists to try and pinpoint where the source is coming from. And very importantly, that we need to partner with industries who are our stakeholders. And of course, we shouldn't end by just saying that this is bad. We need to be able to find alternatives. And so I hope that this could be a point of the discussion during this time in terms of how do we address this problem and how do we find out where these chemicals are coming from, and can we find an alternative to it? Can we provide a means of eliminating these chemicals so that the exposure in humans will be less? Because most of these chemicals are being used in industry, and we use this in our daily lives, and it's almost everywhere, right? So we're exposed to several chemicals, but we need to be mindful of which of how to lower these exposure to humans at the end. So I just only presented these few slides so that we can focus on discussion. And so I just make sure to acknowledge who are part of this team from PGH, the surgery department. I even haven't got a chance to talk about the OBGYN department that we have done some work as well with. And of course, my team at UP, and then also Dr. Roy Gerona and our partner with Dr. Linda Judiz, who introduced Roy and I to this project. Thank you.
All right. Thank you very much, Mike. And I'm an environmental chemist, would be happy to collaborate with you. I've been wanting to do some work with, to link our work with environment and human impact. So I will definitely talk to you about it. So I'm here pleased to introduce Roy Gerona. I know him since we were together as students in UPLB. So it's very nice that he can participate here. He is now an assistant professor in maternal-fetal medicine at the University of California, San Francisco. So Roy, it's your turn.
Let me share my slides. Can you see my slides?
Yes.
Okay. First of all, thanks Diana and Francis for inviting me in this group discussion. Thank you all also for attending this discussion. Good morning and good evening to everyone. So as Diana said, I am a faculty member at UCSF. I direct the Clinical Toxicology and Environmental Biomonitoring Lab and the TB hair analysis lab. In biomonitoring, what we do is we develop methods for endocrine disrupting chemicals, mostly non-persistent organic pollutants. We also do some work on exposomics. We do some work on exposomics in pregnant women, as well as in cohorts that are associated with specific endpoints like breast cancer in women firefighter, and lupus. So I thought that because biomonitoring of endocrine disrupting chemicals is not necessarily currently being done in the Philippines, I prepared really basic slides. So I do apologize for those who are very familiar with the topic. My first two slides, in fact, are very basic. Introduction to biomonitoring and endocrine disrupting chemicals. So with that, I'd start with defining what biomonitoring is. Biomonitoring is the measurement of body burden of toxic environmental chemicals in biological samples, typically blood or urine. An example of a biomonitoring program is the United States National Health and Nutrition Examination Survey, which collect samples from about 5,000 Americans every year, where they measure biomarkers. And starting 1999, they started measuring environmental chemicals as well. Biomonitoring is an integral part in assessing the risk associated with chemicals. And risk assessment is a complex process that has three integral parts. So first, we need to determine the hazard that the chemical causes. What types of effects does a chemical exert in our bodies? And this can be done experimentally by animal studies or in human population by epidemiological studies. We also need to know or understand the dose at which these effects are observed, if there are adverse effects. And the third part is, or the third aspect is human exposure level. We need to figure out how much we are exposed to. And when we do understand the hazard and the dose at which we see adverse effects, then we can determine whether human exposure levels places us in a critical position where we maybe need to clamp down and create regulations to limit our exposure to the chemicals. So exposure assessment is done through biomonitoring. So one popular group of chemicals that are biomonitored are endocrine disrupting chemicals. And I think most of you are familiar that endocrine disrupting chemicals are molecular mimics of hormones that are naturally occurring in our bodies. They affect the synthesis of hormones, their secretion, their transport, receptor binding, action, and elimination. Because hormones play critical roles in fetal development and are required in the physiology of organ systems in human adults. EDCs impact sexual development. They impact cognitive and brain development. They may also cause anatomic deformation. An example is cryptorchidism, or undescended testicles, which are being seen more and more now among infants. And they can contribute to a host of pathophysiologies, including diabetes, obesity, metabolic syndrome, and various types of cancer. So what are the sources of EDCs? Unfortunately, a lot of these chemicals are components of materials that make our modern lives easier for the most part. The following are important classes of EDCs. Bisphenols are precursors of polycarbonates and epoxy resins and are found in transparent reusable drinking bottles. They're also found in epoxy resin lining in tin cans that prevent rusting, and even in dental sealants. Other phenols have bactericidal and fungicidal properties like parabens and triclosan, and so they are incorporated in personal care products or non-alcohol based hand sanitizers. And phthalates are plasticizers and emulsifiers, and so they can be components of children's toys and household cleaning products. Perfluorinated alkyl substances are ingredients in making Teflon, which are used as coatings in non-stick pans. There are other classes of EDCs, which include polyaromatic hydrocarbons, polybrominated biphenyls and flame retardants, polychlorinated biphenyls and dioxins, some pesticides and fungicides, and some pharmaceutical agents, particularly non-steroidal anti-inflammatory drugs. So let me now focus on one class of EDCs and how its biomonitoring has helped in their regulation in the United States. So phthalates are plasticizers. That is, they're softening agent that are added to plastic to increase their flexibility, transparency, and durability. They are also used as gelling agents, stabilizers, lubricants, emulsifying agents, and film formers. I forgot to put in. So an example would be dibutyl phthalate, di-2-ethyl hexyl phthalate, and benzyl butyl phthalate. Because of the versatility and the function of these chemicals, there's really no aspect of our lives that do not intersect with phthalates. So where are they typically found? They are typically components of glues and adhesives, translucent PVC pipes, agricultural adjuvants, children's toys, which I've mentioned already, food packaging, household cleaning materials, personal care products, some gym equipment, medical devices, interior coatings in some pills, and even sex toys. So we are exposed to this chemical either by direct exposure or through the environment. Some of these phthalates can leach out from the products containing them, permeating the environment through air, soil, and water, which can really reach our food source, enter our bodies, and affect different organ systems in our bodies. So in the 90s and early 2000s, there was a growing body of data showing association between phthalates and fetal development, as well as a few health endpoints in children and adults. Hence, starting 1999, the CDC has included a number of environmental chemicals in NHANES. The first few years of biomonitoring phthalates, and I'm showing here monobutyl phthalate, which is the primary metabolite of the most common phthalate at that time, is dibutyl phthalate. It showed ubiquitous presence in the American population and at concentrations that are alarming in children who have shown the highest levels of this chemical. Hence, by 2008, so around 2008, the Consumer Protection Improvement Act in the American Congress permanently banned children's toys and other childcare articles containing more than 0.1% of DBP, butyl benzyl phthalate, and DHP. So although the regulation is actually concentrated on childcare articles, that particular regulation raised awareness in the phthalates. And because of that, in the succeeding NHANES, the levels of monobutyl phthalate dropped not only in children, but in the entire American population. So how relevant is biomonitoring these chemicals to the Philippines? So as Mike has already mentioned, in 2017, we started collaborating with this laboratory at UP Diliman. And at the time, with increasing incidence of breast cancer among Filipinas, Michael was curious about the genetic and environmental contributions to this health problem. So to look at the environmental health component, we recruited about 75 women from PGH with breast cancer and 75 age-matched controls. We administered an exposure survey in these women, and we collected both urine and blood samples from them. Those samples are then transported to the United States in my lab, and we analyzed 10 bisphenols and 8 other environmental phenols, 11 phthalate metabolites, and 12 perfluorinated alkyl substances. So 41 EDCs all in all. So Mike has already shown you that there are differences in the levels of some of these EDCs between cancer patients and the control. However, an equally interesting observation is the overall level among all the women in some of these EDCs. So an example of that is monobutyl phthalate. And this is really alarming because in order for me to graph the level of monobutyl phthalate in Filipino women, I have to recalibrate this graph. So you can see here in this graph, really in the United States, there's a trending down in the levels of monobutyl phthalate since 2008. This is what we saw in Filipino women in 2018. The recalibration made it appear that there is not much downward trend in EBP or in MBP levels in the United States population, because the geometric mean we found in Filipino women is 12 times higher compared to the highest geometric mean measured in the US population. We measured 41 EDCs. So is this observation in MBP an exception? Well, not necessarily. So for MEHP, for example, MEHP is the primary metabolite of DHP. It's another popular phthalate, especially in the 90s and early 2000s. The level we found in Filipino women is as high as when this metabolite was initially biomonitored in the United States in 1999. For bisphenol A, we had the same observation. The BPA levels in Filipino women is much higher than the current, well, this is not really current because NHANES is always late, but you see that there's a downward trend. And in fact, the expected trend here is to be even lower than about 1.3 in the entire American population. So the level of the Filipino women is really much higher than the entire American population. And the same is true for PFAS. I'm using perfluorononoic acid here. As you can see, the level observed in Filipino women, again, we're talking about geometric mean here, is almost twice as the highest geometric mean observed in the American population. So Michael has shown you levels in his presentation of comparison between cancer patients and healthy controls. But with the trend that we're seeing overall among these women, I'd like to end my presentation by asking the title of my talk. Is it really time to start biomonitoring in the Philippines? And I'd like to be the judge of that. With that, I'd like to acknowledge some members of my lab who contributed to the development of the methods that are used in this particular presentation. Two specific members of Mike's lab, Joy and Jobriel. I think Jobriel is in the audience, as well as NIEHS and PICARI for the funding. I do hope that we would have a very elaborate discussion on biomonitoring and the possibility of having that in the Philippines. As Michael has mentioned, and even Lynn, we think that it's really the right time to start this particular scientific work in the Philippines, considering the levels that we're seeing in the cohort. What we've shown is a very, very small portion of the population. It's also a cross-sectional data. It would be really interesting to see levels across the Philippines, and maybe if we can monitor it through time, maybe we can come up with potential suggestions on how we can work with all the stakeholders, including industry, in order to address this issue. Thanks for your time.
Thank you, Roy. It's good to see you again, actually, after many, many years. I won't tell the audience how long, but we were also at UPLB at the same time. Again, if you have questions for our discussants, panelists, or just general thoughts, ideas, please go ahead and type them in the chat, and we'll make sure that we get to them. Our next speaker is Dr. Alexander Young. Dr. Young is a professorial fellow, I think the very first one at UP Diliman, at the Institute of Environmental Science and Meteorology. And we're gonna switch gears a little bit, and he's gonna talk about effects on Philippine coral reefs. Go ahead, Alex.
Hi, thank you. I'll be talking about anthropogenic effects on Philippine coral reefs and impacts on Philippine society. Okay, so Philippine coral reefs have one of the highest diversity of coral reef life in the world. So here's an underwater video I took of some coral reefs off the coast of Palawan. And the coral reefs provide habitats that attract all these pretty little fishes, which in turn attracts larger pelagic fishes that provide food for metropolitan Manila. Coral reef fisheries are responsible for 1/4 of the total fish catch in developing nations. Coral reef fisheries contribute up to 67.5 billion pesos, or 1.35 billion US dollars annually to the Philippine economy. Coral reef fisheries provide over 1 million jobs to small-scale Filipino fishers. And the island of Palawan, our study site, provides 45% of the fish supply for metropolitan Manila. And unfortunately, 25 to 50% of Philippine reefs have degraded, and in some locations, 70%. And in 2018, in an unprecedented move, the Philippine government shut down the entire island of Boracay for 6 whole months to let the coast recover. So Philippine coral reefs have the world's highest threat level to extinction from human impact, according to a report in Science. So this is an example of a hard coral that's bleaching. It turns all white. And when it's irreversible like that, they die. The challenge is the lack of suitable metrics to guide policymakers and coastal ecosystem managers. So according to the Philippine country director of the United Nations Development Program, we lack data and a good understanding of the impact we are having on the oceans and how much we depend on them for our own survival. The Philippine government reported to the United States nations about the lack of measurement methods for a number of sustainable development goal targets, including Goal 14, conserve and sustainably use the oceans, seas, and marine resources for sustainable development. So you can see that there's an urgent need for superior metrics in coral disease diagnostics, which we're trying to develop by creating an early warning system for coral bleaching, by looking at changes in gene activity induced by environmental stress that leads to coral bleaching. And if we're able to develop a coral reef molecular diagnostic that can pinpoint point sources of pollution, this can guide policymakers to recommend policy changes and enact laws to enforce policy changes. So by identifying stress-specific biomarkers that can pinpoint specific sources of environmental pollutants before rather than after irreparable damage to reefs occur, we hope to create a early warning system for coral bleaching. So we decided to look at and track clownfish hosting sea anemones, which also bleach, as you can see in this photograph that I took. And by tracking clownfish hosting sea anemones, which are much easier to do underwater during scientific scuba dives than with hard coral, we can easily GPS track statistically significant numbers to do quantitative geospatial mapping studies. So we first looked at petroleum hydrocarbon pollution. Sources of pollution by hydrocarbons include storm runoff from urban areas, low traffic, fuel depots. And we measured petroleum hydrocarbon in Puerto Princesa Bay using an instrument that optically measures something called BTEX, which stands for benzene, toluene, ethylbenzene, and xylene, which are the degradation products of petroleum hydrocarbons. And MCL is the maximum contaminated level tolerated by the US EPA in drinking water. So here's some examples of fuel tanks that line Puerto Princesa Bay, which provides all the fuel for the entire island, as well as for all the jets that land at Puerto Princesa Airport. And these fishing communities are known to dump used oil directly into the bay. So when we measure BTEX hydrocarbons throughout Puerto Princesa Bay, this is the water and this is the land, we detect 4 hotspots where red indicates high levels of BTEX and then blue indicates low. You can actually see the petroleum hydrocarbons floating on the water from the air. We geospatially mapped through underwater GPS mapping the location and the health of clownfish hosting sea anemones encountered during reconnaissance scuba dives, where the black crosses represent healthy sea anemones and the white crosses represent bleached sea anemones. And because we have such a high resolution GPS tracking underwater, we're able to quantitate the amount of bleaching within each contour level. So here you can see that below 20 parts per million, we detected no bleached sea anemones in this small pilot study of 147 sea anemones. But at the highest concentrations, we detect 60% bleaching. So this is pretty good evidence that the amount of hydrocarbon pollution that we detect at Puerto Princesa Bay is associated with bleaching. So what we want to do is use a translational research approach by simultaneously combining field studies with controlled laboratory studies to identify petroleum-specific biomarkers. And here, this is just to show that we are able to purify DNA and RNA biomarkers in coral and sea anemones detected by PCR, RNA for transcriptomics, bacterial DNA biomarkers, and viral biomarkers. So by identifying petroleum-specific biomarkers by transcriptomic analysis, followed by differentiate RNA analysis, we hope to eventually develop a molecular coral disease diagnostic. We can get clues from oil and eating bacteria and metabolic pathways for degrading BTEX, where the green boxes represent genes that code for enzymes that metabolize BTEX. We can also get clues from eukaryotic and human systems in metabolic pathways that degrade polycyclic or metabolize polycyclic aromatic hydrocarbons. And interestingly, some of these metabolites, the epoxides, react directly with DNA and can cause cancer, which has implications for the potential health consequences to humans from bioaccumulation of polycyclic aromatic hydrocarbons in fish. So in conclusion, coral reefs are essential to the Philippine economy. We detected high levels of petroleum hydrocarbons in Puerto Princesa Bay and associated it with bleaching. There's an urgent need for superior metrics and molecular coral disease diagnostics to guide policymakers and coastal ecosystem managers. And we recommend policy to create a national program to develop actionable environmental metrics to guide policymakers and ecosystem managers in protecting Philippine biodiversity and human health. Thank you.
Thank you, Alex. I think we should also know that Alex actually is American and from the US and moved to the Philippines. So again, he's one of those academics who moved to the Philippines because of the biodiversity in the Philippines. Okay, our last discussant is Lara Ortega Ibanez, and she is the country director of the organization Pure Earth Philippines. So Lara, you have the floor. Lara, I think you're on mute.
We can't hear you, Lara. Yeah.
We can see your slide, but we can't hear you.
Hello.
Now we can hear you.
Okay.
All right. Okay. Good morning, everyone. Thank you, Doc Francis and Doc Diana, for inviting me to be part of this forum or meeting. As I said during our meeting, it's intimidating to be part of this scientific community, but actually I love hearing from all the panelists, and I'd love to be part of this because my hopes are up again that we can get things moving when it comes to environmental pollution and health. So my presentation will be about strategic interventions in the pollution pathway and population framework. And yes, I am from Pure Earth. It's an international nonprofit organization based in New York, but with operations in about 20 countries, focusing on reducing toxic pollution in the low and middle income countries, especially as it impacts public health. So if you want to know more about our organization, you can check out pureearth.org. And we're also part of the Global Alliance on Health and Pollution, the Lancet report that Dr. Lynn presented earlier. Pure Earth was part of that. And to give you a snapshot of what we have been doing in the Philippines, basically I can summarize it into three things. We do action research. We are a nonprofit whose focus is not really more on advocacy, but more on research and science-based interventions. And here we do initial site screenings, mostly of soil contaminated sites. And we do also some detailed site assessments where we have provisions for that. Blood lead level assessments, again, where we only do blood lead level assessments when we have funding for a proposed cleanup. And we do some exposure mitigation as well as water, soil, and fish monitoring. So I would have wanted to have met all of our speakers earlier so that we could have collaborated on a number of fronts for the past 8 years. And then we do awareness raising, basically on three major stakeholder groups: government, on policies, benchmarks, data sharing on our identified sites, findings, impacts, and also potential solutions. And then we also touch base with the private sector in the industry as much as possible. As Doc Michael said, we hope to, we touch base with both the formal and the informal industry players and talk about benchmarks and alternatives wherever possible, and a community where we do capacity awareness raising and at the same time capacity building, where we train investigators for our site screenings, trainings for the local environment and health personnel. We do policy drafts for the local governments, guidance documents, technical assistance, and all those sorts of things. Our framework basically is actually pollution, migration, pathway, and then the people. But for the sake of presentation, I'll just use 3 Ps: pollution, and then the pathway, and the population. So pollution, as we all know, we'll be looking at the sources of pollution. It could be the source, the nature, the concentration levels of the toxicants present from those sources, some of which have already been mentioned by our previous speakers. And then the migration between the source and the potential receptor community, and some pathways, as Dr. Lynn mentioned, where from the migration routes are there ways for these toxicants to enter into the human body. And the people or the population, the impacted community. This is the flagship program of Pure Earth worldwide, Toxic Sites Identification Program, where we do targeted site assessments for sites where there is a known source of pollution. But here in the Philippines, again, it has expanded to a variety of things, where we did some targeted assessments before, and just recently, we also did investigative site assessments where we don't have any knowledge about the sources of pollution in those particular cities, but we blanketed all the barangays of those cities and check out the levels in soil. Again, for the Philippines, we have touched base on some water monitoring and fish monitoring, which I will touch on later on. Okay, so for the pollution part, these are basically more of proposals based on our project experiences. First is area-based baseline studies and action planning. Because we have attended a lot of all these stakeholder meetings with the government and all other stakeholders, and sometimes there's a plan and there are activities that are reported every quarter and so on, but there's no checking as to whether the activities are really appropriate for the issues being tackled. And so this map here for the area-based baseline studies, we did that for one project where I did all the establishments in that particular area near a legacy site of a used lead acid battery recycling facility, and checked out how it will impact you know, contamination and health and all that and all those. And strengthen science-based interventions and evaluations. Again, I would relate this to our river quality monitoring database, which we developed and actually proposed to the government to be used for the WACMAS. If you talk about water quality management areas in the Philippines, these are the river systems where that are considered, you know, hotspots of contamination, and that the stakeholders, the government and the private sector, as well as the nonprofits, have to go together and come up with an action plan to deal with the pollution sources and the impacts. What we did was to develop a database where you can see on the left side, I guess, the red spots. Our proposal is if you see the sampling points, it will automatically be red if it's exceeding the standards. And our proposal was that from those red points through GIS, you can develop like a radius or model a radius for the potential sources of why we have that kind of pollutant there. And at the same time, identify if there's a prospective community that will be impacted by such levels. On the other side, the strengthened science-based interventions evaluation, this is just a sample graph of lead in sediments in a water system, in a river system, where we have done monitoring consistently for 2014, 2016 at least, and even before that, where you see that actually the level is down there, and almost all the sampling points are exceeding in terms of the levels. Okay, so for the pathway or the migration routes, our proposal here is to really do multi-partite monitoring and studies. Again, in our projects before, for the monitoring part, we actually involved training local government representatives and also volunteers, for instance, from HSBC, our funder for one program, their employees and some community representatives, so that they know, you know, how to test their water and know what's the state of their water systems. The thing is, we do have monitoring from the government, right? But the data we found out is usually just shared among the agencies, and sometimes even the local government units don't know the results of this monitoring. And so the beauty of multi-partite is that everyone who's concerned and involved will know the status of the river system or the water system, and do what they have to do. And then next is appropriate information sharing platforms, such as this one. This is actually Dr. Lynn, Panganiban. But this is something that is sensitive, as Dr. Michael said earlier, because when you talk about pollutants and the levels of pollutants, then there are a lot of people who might be, and industries that might be impacted by this information. So it has to be, again, a meeting of the minds as to what information do we need to share among ourselves, in the scientific community, in the government for policy, and then what is for the public, so that they can also do something to mitigate their exposures. And then as a specific action point, I put in there certification of local fish farming inputs, process, and produce. Again, because part of our project experience is to work into fish farming ponds, the source of which is a polluted river system. And so our learning there, it's so costly really to solve pollution downstream. So I'll go back to the first slide, that it's still most effective to solve pollution at source. But while that is a long shot, we should do something when it comes to the migration routes and the pathways. And aquaculture is something, Philippines being, I think, one of the leading in terms of aquaculture in Asia. if we do certification actually when we export our products, right? Because the receiving country will have to impose their standards. But for local consumption, we are not as stringent when it comes to what kind of inputs. The feeds, for instance, we have found out before that the feeds have heavy metals in them. Even the fingerlings that we stock in our ponds, the process and also the produce actually before they end up in our markets, are they really safe to be eaten or to be consumed by the public? And then actually here with Dr. Lynn Panganiban, we developed just a localized advisory in a certain barangay where we have found heavy metals in fish. You know, the type of like you can just eat this amount of fish in a week so that your exposure is not something that will really adversely impact your health. Okay, so for the last part, population or the people, I think this is where we need to really, you know, put our minds together and hands together, because from experience, from Pure Earth, we've been working on this for like 10 years or more. And we've been trying to do something from the end of, from the pollution sources to the pathways or from the environmental perspective. But I believe unless we put it in the health perspective and show, you know, the decision makers that this is already affecting your people and your future and the children, then action will not be taken and it will not be prioritized among all other concerns for low and middle income countries like the Philippines. Environmental pollution or sometimes always gets, you know, down the line. But if you talk about public health, then it's automatically prioritized. So for this part, I resonate with all what the speakers have mentioned earlier. Local environmental risk factors and health correlation studies. We really have to generate more local data, because sometimes, you know, if you present it to decision makers, they'd say that it's from another country. But so we have to generate more local data when it comes to environmental risk factors and health correlation studies. And then the capacitation of personnel capacitation in terms of personnel, for laboratories and surveillance and response systems. Dr. Lynn, I believe, touched on this a while ago. The thing is, as we have discussed, the impact of environmental pollution is mostly on low and middle income countries, and the most vulnerable populations are the children. And then the proactive interventions would be capacitation, not only for us to be able to do the studies, but for us to be able to act on our findings. I think that's the most important link here. And specifically as an action point, I'd like to propose blood lead level baseline assessment for children. That's why I'm so interested with the work of Kasia as well, because in our toxic sites identification program, actually mercury and lead are the top toxicants that we saw. For lead, for instance, the sources, Dr. Lynn, I saw enumerated already in the chat box: paints, ULAB recycling, cosmetics and toys. And in one area that we worked on, the BLL results showed that 100% of the children had over 20 micrograms per deciliter. And actually 46% of them above what our test kits can measure, which is above 65 micrograms per deciliter. And that's just one site. We don't even know the whole picture nationwide. But the Institute of Health Metrics and Evaluation says that approximately 20 million children in the Philippines are exposed to lead. We are in the top 20% of over 200 plus countries with lead exposures. I asked actually how they come up with such a projection, and it's using some of the previous studies with BLL studies to do some modeling. But again, if you present this to decision makers, they'd say we need, you know, locally generated data. And I'd like to relate this not only to children's health, but even to education, because as you said, it impacts the IQ. And for instance, the recent one in the PISA report, right? We were like the the lowest performing when it comes to just comprehension and literacy. And so I'm actually thinking if there's a correlation with the environmental risk factors, and not only with the health endpoints, but also with the education performance of our country. So with that, I want to thank you and again acknowledge also our donors for the past 8 years, the HSBC Water Program, UNIDO, USAID, and ADB, and as well as UNDP GEF. Thank you once again. I hope that showed you at least a part and a snapshot of what we're doing on the ground.
Thank you, Lara, for this very nice presentation. And we have a lot of questions here. Some have been answered, but we will read some of them that have not been answered. Obviously, it was really obvious that there's a lot of monitoring going on, but there's not coordinated monitoring on environment and also on humans. There's a question from Dr. Safarale that hasn't been answered. How far has the research gone on the endocrine disruptors in Laguna Lake? I know there's a lot of people working on Laguna Lake, but there's like bits and pieces. Is anybody in the audience familiar with an ongoing monitoring program in Laguna Lake? And if you answer, please make sure you unmute yourself so everybody can hear it. No one?
Diana, I thought Pythias Espino, Dr. Espino from UP Diliman is doing that. Do you know her research at all?
Yeah, she is. She's not here today, but I haven't seen anything that's like published long term. So I don't think that there has been long term monitoring on endocrine disrupting chemicals. I think part of it has been mentioned. Part of it is a lot of this analysis is done using LCMS, which is very tricky, very expensive, or if you have it, it's very difficult to maintain. I know for sure that in UP Diliman, there's a couple of LCMS, but once it's broken, it's hard to fix. It's been a problem, challenging even for us. So that's one of the problems here. Francis, do you have something that you selected? We were just gonna alternate questions. And if anybody hasn't typed their questions here, feel free to unmute yourself and just speak out.
Or if you want, you can use the icon, just raise your hand and we'll call on you. It's a small group, so I think we can, we can have people just chime in. There's actually a nice comment from Michael Promentilia. Maybe Michael, you can read your comment or expound on it. Or maybe I'll just read it. Basically, his point is that obviously he agrees that the risk assessment process is important, but sometimes we overlook the risk management part, including risk communication. Our regulatory and implementing agencies should also be capacitated in this aspect. And this may be one of the reasons why agencies are adamant to share or adamant to not share data or share data. Yeah, lack of data is also another problem. Data problem is a social problem. Are there any suggestions on how we should address such issues as well? So there are two aspects to that. One is the risk communication and risk management. And then the other thing is just how we generate and share the data. Any comments from any of our discussants? Go ahead, Lynn. Yeah.
Thank you for your comment. Very much appreciated. Well, all of us are doing a lot of work in establishing risk assessment, but really, truly, when it comes to managing the risk and communicating is something that we really need to emphasize on. In the Philippines, actually, we do have committees and bodies with the purpose of addressing this. So I would think in place of the Interagency Committee on Environmental Health. This is a committee that is supposedly composed of top-level leaders in the different agencies, but often those who come to the meeting would be, of course, the middle managers. But rest assured there is actually already a system in place, but what is maybe at this time that we need to improve on is to really keep that committee working, addressing problems that are facing us. I used to sit in that committee because the academe is represented and the National Poison Management and Control Center has been identified as one of those sitting in the committee, especially on toxic substances. But I would only just experience it that when there is a disaster that happens, and that may be in relation to chemical whatsoever, that's when the committee convenes and then act on it. So we're more of a reactionary thing rather than proactive. I know in the 90s there were efforts of really doing it. The big committee divided the different agencies of government into smaller subcommittees with the aim of really working on this end in managing the risk. But somewhere along the line, maybe because of other pressing priorities in the country, it never materialized. So who heads this meeting, this committee? It's actually the Department of Health who heads it, and the Department of Environmental and Natural Resources. DNR is the like the vice chair of that committee, and the secretariat is the Department of Health. But I think with the pressing concerns right now, I don't know, it has been years now since that committee has come up with. There is also a National Environment Health Action Plan, or the NEHAP. This is, I think, as part of our commitment to environmental concerns as far as international organization is concerned. But the last NEHAP that was created, I don't know, Lara might be able to share more, it was sometime in 2018 when we had to look at all our initiatives and where are we right now. And it's very encompassing, from biomonitoring, environmental, health effects, etc. But really very good programs, but you're correct, it's something like we really need to come up and complete the whole cycle to really be able to address it from source to the population at risk, and doing something as far as remediation is concerned.
So Lynn, those are really good information to know. But so what are strategies by these agencies and organizations in order to make, say, risk assessment data and risk management strategies accessible to the general public? Because obviously, you know, the organizations can put up the data and come up with strategies, but all of that will be in vain unless the public really knows what's going on and how they can do their part in the risk management strategy. So is there an organization? Are there third parties involved? For example, here at UCSF, the Program on Reproductive Health and the Environment actually puts risk management protocols and assessment in a language that can be understood by ordinary pregnant women. And how does that apply to their specific situation? So are there departments at UP or other academic institutions that also do that? Or
by my experience, we usually have the private organizations or non-government organizations who will help out and reach out in the community level. The government still embarks on the involvement of local government agencies, but as we know, not everybody's really has the expertise or the skill on really be able to translate what is a technical matter down to a level that is understandable, and making people really own that particular action point. With Pure Earth, we've been doing that a lot of times, really involving the people in the community and making them aware in terms of alerting their local government agencies. But it takes a lot of effort. When it comes to the government, there's really none that really will engage itself on that particular purpose. No, the system in the Department of Health it's always been there in terms of that office there, but really and enforcing somebody to be doing that is something that is not yet there. And as I've said, there are data, there are information. It's just that maybe there's the lack of the capacity on how to translate it. And so therefore, I think what Michael was saying is that we have data, but how do we translate that in a way that we're not also trying to put down and cause some industries not to compromise the products of industries. And at the same time, we also want to protect our community, the people in the community. So there has to be that part where we really need somebody to be doing at that on a level. As scientists or people in the academe, we can provide the data, but we need somebody else to be doing that and helping us out in translating what we do have in terms of policy and eventually in terms of really ensuring that the people take responsibility in protecting their health and protecting the environment.
So then our social scientists and communications expert
Yes.
are integral part of these organizations and committees, because it seems like that's critical in translating scientific data from the scientists and engineers to the public.
Right.
Very, very important.
Dr. Roy and Dr. Lynn, can I also add, I guess, from experience also, right? Before, we actually proposed to, say, the government, to have like a website for the public, but actually in a positive note, like report what we are doing, like all the sectors, what we are doing to address pollution. So that, because sometimes, people would eventually always worry about if we put out data in the public and there will be reactions and we're not ready to address it. But to make it like a positive thing, that we are just reporting, this is what we're doing. We're not just putting out the problem, but this is what we're doing about the problem. But the thing is, as Dr. Lynn said, I think we still have a lot to mature on when it comes to that kind of communicating. For instance, one example is in that particular local government where we worked on, we tried to be as transparent as we can. So every time we finish our sampling and results, we share it to the local government. But the thing is, it went out to a newspaper company, and then they cited just Pure Earth, and no one would like to admit who gave the information, and the industry took it against us, that instead of, it's counterproductive, because as Michael said, the industry allows us to take samples and give recommendations as to how they can better their operations and all that. So if we don't handle the communication part, it can be counterproductive at some points. It's something that really we need to come up with, like a strong and clear series of action points when it comes to risk communication.
May I say something?
Thank you, Lara.
Yeah, I agree
go ahead,
with that.
Alex. Yeah.
Yeah, I agree that communication is the key issue in terms of implementing policy. One, I think that the politicians need to learn a lot more science. I mean, that's clear to me. And number two, I think the scientist needs to learn how to communicate effectively to the politicians. So I keep thinking about this Yale study where they took the example of the Space Shuttle Challenger accident. You remember that blew up because they decided to launch in cold weather and the O-rings contracted. And there were actually memos written by the engineers to the top management warning them not to launch. But the message got lost in technical jargon that the administrators didn't even understand the key point of the message, which was not to launch, otherwise it may explode. The other point I want to make is, so I'm a United States citizen and recently came to the Philippines, and one thing that sort of surprised me is what people are talking about, how they're afraid to disclose data because they're afraid of backlash. When I was in Palawan during these studies, the environmentalists actually got assassinated by the governor of Palawan because he was making complaints. So that's a whole other twist that I have to adjust to. So I don't know, is that actually a big issue here? Like, when I hear people are afraid of identifying themselves as who collected the data because they're afraid of backlash by industry, or I guess that's a question of mine.
Yeah, sorry, I guess you're going in and out, Alex, but I think the question was about backlash from revealing or sharing data.
Yeah.
Can anybody, I mean, I think
Well, I
obviously.
can understand it because I was originally in biomedical, biotechnology and pharmaceutical industry. And when we were developing new molecular diagnostics for toxicology, we actually I was surprised how a lot of people in the toxicology departments didn't want us to develop something that the FDA could use against them. But I think now they're all on board, so it's sort of a learning curve to implement new technologies for environmental sciences.
Yeah, so I guess that's an open question to to everybody, and maybe some of our discussants, if you want to address that, especially those working in the Philippines. Michael or Lynn or Lara.
Yeah, actually, can I speak, Francis?
Sure, go ahead, yeah.
So my worries actually first is the institutional framework we have. So perhaps some of our resource speakers could help us in this aspect because, for example, this is a purview of DOH and DNR or EMB DNR in terms of if you're looking at environmental health, but I'm not sure whether they are talking with each other as regard to this kind of policies that will help us really, how would say, to have this kind of initiative. I think it's, even science and even in the academy, we are really mainstreaming this kind of environmental health. Sad to say, we still lack the capacity to do this kind of research because of course, of some problems with instruments, and that's why we are working with other collaborators from the US and so on. So, but still, in terms of policy, we still have to really, how to say, find a way to solve this kind of a problem. So, what do you think about this one?
Go ahead, Lynn. Yeah.
Can I give a comment?
Yeah.
Efforts are being made. I'm very just hopeful about our government, but there are a lot of efforts being made by the different agencies. But the problem is we work in silos. Okay, so when there are opportunities for us to be sharing, these are very, very few. So I mentioned about the Interagency Committee on Environmental Health. There aren't really opportunities for governments to really gel and work together because everybody's coming from a different point of view. Just to cite a point, when there were disasters that happened, and now we have to set levels, remediation levels, or we have to suggest what would be a level that we need to aim at in terms of environment and biologic effects. The government agencies would spend some time in really arguing of what would be the level. So I clearly remember that when there was an accident in one school here and there was a mercury exposure to children, and we're trying to set remediation goals, etc. And one agency would say this is our level, and the health agency would say this is the level. Occupational health again, because there's another agency doing that. So we're all working in our own silos. We're all working within our own government, but again, opportunities for us to meet and really be on the same mind and with the same purpose. We are still in that area where I think we're still a work in progress. I would say so.
And also, if I may add, Michael, on that, I think in terms of policies, we are way advanced when it comes to, like, in the Asian region, we have the policies in place. But the thing is, policy presence does not mean that it's also policy implemented. I think that's where the problem lies. And And as Dr. Lynn said, when it comes to implementation, that's where the silos come in. Like for instance, in our pond project, when we're talking about water inside the ponds, we have to talk with BIFAR. But if we're talking about the water outside the ponds, then we have to talk with DNR. And so, you know, all these things. And then leveling, like, for instance, the national government would say, we have the policies in place. It's up to the local government now to implement. And then the local government would say, yes, the policy is in place, but we are not capacitated to understand and to implement the policy. So those kinds of, you know, struggles on the ground are happening.
Thank you, Lara. So sorry, Diana, do you want to go ahead and
Ask Caro to speak, but I think it's the same thing you're gonna do, right?
Yeah. So I had asked Caroline Haraula. She's a professor at UP Diliman, to maybe share some of her data. I think they also work on this and how they have some data on groundwater. So go ahead, Caro.
You're muted, Caro. Caro, you're muted. We can't hear you. Okay.
Hello. Yeah. Thank you very much for organizing this group discussion. It's really very helpful. We come in from the oceanography side, marine geology, and in terms of policy or environmental health and monitoring, it would also be great to incorporate factors like the bedrock. Like for most of we are archipelagic country, and there's a lot of our islands that are mainly limestone, carbonate, sandstone, very porous to groundwater, which act as conduits of all these emerging and persistent organic pollutants, as well as trace metals. In some areas, we have hydrothermal systems, such as those in Laguna, Batangas. You see a lot of hot springs, but the pH and temperature and salinity in these areas can also redissolve some of the metals, and some high trace metals could be released from this area. So in one study area in Batangas, for example, there is slightly high arsenic, and we were already expecting that because it's a hydrothermal system coming out from terrestrial going into the coral reef areas. So I mean, these things would be nice to monitor and consider in all our monitoring efforts, the release of submarine groundwater discharge. So in Bulacan, Pampanga area, for example, it's also porous, made up of sediments, but with sea level rise, we have salt water coming in. And with the change in DO salinity, a lot of metals could be redissolved or precipitated, and this can also be a huge effect. So I could share very quickly what we've learned from Boracay in terms of wastewater dispersal, and what policies we could formulate from those.
Yes, go ahead. You want to share your screen? If you have, are you going to show data? Or
okay.
But, yeah.
Let me see. I'll share screen. We collected water samples from around the island. Some of the analysis we did, we collaborated with Dr. Diana Aga. These were analyzed in her laboratory. For example, caffeine, there's no cocoa or coffee plantation in Boracay, so caffeine is a good tracer for wastewater. Where you find way offshore at OM8, we already find residues of caffeine, so that probably comes from wastewater discharge. If you trace it back on shore, if you squint your eyes a little bit, you'll see these two lines, very faint trace. Those are your wastewater discharge pipes. There are several that we could trace around the island. There are shifts since the closure For example, this is caffeine. During closure, it's color beige. When it opened up around 2019, when we got back there again, it's huge shoot up in caffeine concentrations of these waters. There are also acetaminophen. In some places, the highest concentration is in this coastal reservoir right here, where it's surrounded by a lot of houses. So for example, here where the wastewater also discharges, you'll find higher concentrations of caffeine as well. So using these emerging pollutants as tracers, as well as the more traditional ways of tracing them, such as wastewater discharge would be fresh water. It might contain E. coli, putting them all together in a model. This is a big project, and I collaborate as well with Dr. Villanoy on this from UPMSI. So his group also used to model the dispersal. On side, we have the bottom waters. On the right side, these are surface. We have hypothetical discharge pipes in 3 sites on the eastern side of the island. So some of the recommendations that we would like to give the local government is, for example, there's very quick dilution dispersal on the northern and southern sides, but there's stagnant water and very slow dispersal on the middle of the island. So in terms of where you put the wastewater pipes, it would be good that they're more concentrated in northern southern sides. It's also best to consider tidal fluctuation. It's also best to consider the length of the pipes, where they're gonna go. So these are some of the items that we've learned from our study with Boracay, but Boracay is just one example. A lot of other islands in our archipelago have similar bedrock as Boracay, limestone and sandstones, very porous. It has faults cutting through it, joints cutting through it. So I think in terms of putting it all in perspective, perhaps it would be good to also include the geology of the area, submarine groundwater discharge, and the general coastal morphology of the area.
Thank you, Caro. Diana?
Yeah, I actually, well, we have a question, some thoughts from George, but I actually wanted to ask Kasia. I hope, are you still here, Kasia? I think you are.
Yes,
yeah, because so a lot of these discussions, of course, all the research need funding, right? And Philippine funding is very limited, as we all know, but there are a lot of other funding agencies that are national, international, global, and even based in the US. And I wonder, so NIH, which is based in US, they fund studies that are not in the US. What is, I guess, how do you go about that? Do you always need the PI to be from the US? And how do you convince NIH from the US to fund a study outside the US? I guess that's.
That's a tough question, and I think things have changed even in the last couple of years. So, you know, my experience has been in Uruguay. We've since 2009 have had NIH funding from the National Institutes of Environmental Health, and that originally began through a program that was specifically aimed at doing research in low and middle income countries. So we were able to get an R21 through that mechanism, which later opened up an opportunity to apply for further funds through the NIH to continue with that work. But even outside of that program, we've had once sort of the pilot work laid the ground to establish our research program, then we've been able to go back and apply. So it is not necessary to have a US partner. Many of the calls are open to foreign institutions. The biggest hurdle to jump through with the NIH is to demonstrate that the research cannot be done elsewhere, or specifically that it cannot be done in the United States. So if that can be demonstrated, then funding could be available to institutions outside of the United States. Initially, when review sections look at a research idea, they are mostly interested in funding, or the review criteria are that the problem needs to be significant, that the approach is innovative, and that the approach can actually be accomplished in a, as proposed and in a given period of time. And then there are specific discussions of whether the research study can, or whether there's sort of the bar has been met that the research study has to be conducted outside of the United States. So it's possible, but it's not easy. And I think that probably when scientific review committees make decisions as to whether a study is fundable or not, they of course cannot take into consideration whether it should be funded. There is a different level of scrutiny through the councils that determines whether funding will be made for a specific proposal. And I think at that level there may be other considerations that I'm just not privy to. So I can't speak to them. So outside of the United States, there's funding available, especially for people who have connections to European colleagues through the European Research Council. Wellcome Trust is interested in funding research outside of the United States. And on specific issues, the Gates Foundation, although admittedly, I haven't applied very many times to the Gates Foundation, but through their grant challenges, I don't know to what extent they would be interested in funding environmental issues. To my read, and again, I haven't really applied very much, so I may be completely wrong, but to my read, they're more interested in infectious diseases or other types of questions or issues that are not very environmental, but others may have other perspectives.
I think it's definitely possible. I mean, the NIH gave $3 million to Zhengli Shi in China to conduct coronavirus research and the environmental impacts of zoonotic transmission from animals to humans until Trump terminated that relationship.
Right. I think that's a very specific strategic funding that's made that's of interest to the United States. I don't know to what extent environmental issues.
Yeah, we could ask. We actually have a PAASE member and former president, I think, of PAASE, who's an NIH program director.
There you go.
So I mean, we can ask. This is Leah Croucher. Yeah. We have a few more minutes. We still have some time, but I wanted to ask this question of the panelists and give you some time to think about this, because we're talking about all of these issues and we heard each other and all the comments, and there's a lot going on in the chat, and that's going to be recorded also. So if it's on the chat, even if we didn't get to your question or comment, please know that that's going to be recorded and will also inform our recommendations. But the question for the discussants, which I'll give you some time to think about this, and we'll ask some people first, really is to think about, recommendations, things that you almost like a wish list, like if, thinking about what we can put together as PAASE in front of the Department of Science and Technology. So from your standpoint and having heard everybody discuss, are you advocating for, one or two things that you think the government should do or the DOST should put together? Okay, so I'll give you a few minutes to think about that. But I also wanted to ask a few people. We have some experts also who are not discussants, but they're here in the audience. I think the first one is, so let me just identify the people who I recognize may be able to contribute to this discussion. Dr. Veronica Amigo, I think Dr. Lorely Trinidad, I think academician Virginia Cuevas was here earlier, but she left. Maybe Dr. Nina Cadiz. I don't know if Nina has some thoughts about this, but so just think about that, and we'll call on George Emmanuel. I think he's got some thoughts he wants to share.
Thanks, Francis. I just wanted to raise some issues on the issue of the risk assessment methodology. It's a much bigger issue, but I think we need to face it and find ways to deal with it internationally. This is connected to perhaps the most recent research I was involved in towards the end of last year, and that is on the impact of food contact chemicals in plastics. But the issue is really much broader. One is, as we do risk assessment, we found that one of the issues is that it's very difficult to get, especially from industry, a complete list of all the substances that are being placed in my particular case, in plastics used for food contact. We later found out, of course, that there are hundreds of these chemicals. So when you're told polyethylene or polypropylene, it's not pure. There's actually dozens and dozens of chemicals added. Some were mentioned, of course, phthalates and so on. But there's many more that I wasn't even aware of until I got more into it. So that's one issue, getting information on what are those substances. The second problem is that most of those substances have very little studies in them. So we don't know what their health impacts are. We know very little about many of the other substances. In the case of the US, many of them had just been passed through because of GRAS, generally recognized as safe compounds. Fortunately, in Europe, the CLEAN program is identifying more and more of substances of concern. That's one issue. The other issue is we're dealing with mixture toxicity. So we have many substances and how they impact people who are exposed, the synergism between the different compounds. So that's an issue. Another issue has to do with how we develop exposure. So for example, even for a single chemical, there may be multiple, and there are often multiple exposure sources, and how do we deal with that? And so all of this falls down into the issue of thresholds. What thresholds then make sense? Many times thresholds are decided on a substance by substance basis, and yet we're dealing with mixture toxicities. There are issues of, for example, how do we determine thresholds of substances with non-monotonic dose response characteristics. And of course, the vast majority of the substances have no thresholds at all. So I think these are all the challenges we're faced as we're trying to find ways to protect the public more and more from exposures to all the chemicals that we're now faced with in our society. That's basically the gist of the comments I wanted to make, because I think we shouldn't assume that existing risk assessment methodologies are the final say. It's something that needs to continue to evolve as we understand more and more about all these other chemicals.
Thank you, George.
Can I add something to that?
Sure, go ahead, Roy.
So another, I think this is something that I think not a lot are aware of. When industry defines certain terms, we're so used to, in chemistry, we're so used to having the term being accurate in its meaning. So an example of that is diisononyl phthalate, right? So when we think about isononyl phthalate, we think about the branch form of a 9-carbon based phthalate, right? And that iso means that the branch point is at the second to the last carbon all the time in organic chemistry. But industry loosely defines iso. So and what is the implication of that? Well, the implication of that is there are so many different possibilities of a branch structure that is iso. And that all of those possibilities are loosely named iso. So when even if you ask, okay, how much isononyl phthalate is in this compound, you don't really know which iso structure are they referring to. So that's one. The other is the implication of that in biomonitoring. When you biomonitor any given chemical, there are many different pathways by which that particular compound, especially the phthalates, can go through. And so the question is, how do you assess the overall exposure level of an individual to an isononyl phthalate? Well, for isononyl phthalate, it's really impossible because one, you're not talking about a single chemical entity. You're talking about several different chemical entities that could potentially have multiple different metabolites. And so when we think about biomonitoring for a lot of these newer chemicals that are coming out, we need to always bear that in mind, because I feel like, there has been, at one point, there has been a question in some of the data that we're generating for isononyl phthalate and why the data that's coming from our lab is not coinciding with data from European labs. And when we did a little bit more search on the literature, well, we found out that really will not be able to find the metabolites that the Europeans are looking for, because the products and the chemicals, although they are referred to as the same, are very different. And so that further complicates this issue in mixtures, because aside from the fact that industries can actually pick and choose which they have or they can put in a product, they can also have multiple possibilities for a single name for a chemical entity.
Thank you, Roy. We have another question or maybe comment from Dr. Baha. And then after that, we will try to wrap up. We don't want to go past 10:30, but Dr. Baha, could you please share your thoughts?
Hi, thank you for this webinar. I've learned a lot. I think my comments would be, first, there's a lot of expertise in terms of the exposure assessment side of water pollution in the Philippines. We have chemists, we have geologists, and all of those. But I think what really needs to be improved right now is doing epidemiological studies relating health effects of being exposed to these chemicals and heavy metals. And I think PAASE would be a good vehicle for collaborative work among exposure assessment experts, scientists, and epidemiologists. But unfortunately, we are a limited species in the Philippines. I know there's not a lot of epidemiologists in the Philippines right now, and I'm an environmental epidemiology looking at air pollution, but would now, hopefully, if funding works, would look at water pollution also, water epidemiology. So again, thank you to Francis and Diana for this webinar, and I hope we can do more collaborative work and looking at cohort studies and looking at the effects of heavy metal exposure or chemicals longitudinally to at-risk populations. Thank you.
Thank you, Mariel. So before I turn this to Francis, I'd like to take this opportunity to thank all our speakers and all the participants who attended and all the people that made comments and chatted. But what I take from this is we really need to start thinking about connecting what is in our water and what impacts do they have to humans. And that's actually been something that I've always wanted to do. And so I've been lately collaborating with Kasia, who is an epidemiologist, because we as chemists, we can measure anything, but without connecting them to the people that are affected, it's meaningless. So, I mean, that's part of the thing that I wanted to get into this. And so we've tackled several things, you know, environment, also communicating to people, and basically biomonitoring is something that's really missing in the Philippines. But of course, one of the challenges, again, the analytical chemistry is not very easy and not cheap, right? So we need all this funding. And so the government, we should encourage the government to think about funding a lot of this work. And I think one of the things that also should come out with it is coordinated studies is very important. We all have our own little different topics, and you know, but we need to work together to really make it more meaningful. And with that, I'd like to turn this back to Francis.
So thank you, Diana. I promised that I would ask this question of the panelists, of the discussants, the speakers. If you could just 1 minute each, just one last chance to just basically say, you know, maybe your recommendation, your final thoughts. I mean, if we're going to put something together that we're going to push to the OST, for example, what would be your recommendation? Maybe we can do this in the order that you spoke. So let's start with, so Diana already went, so Kasia.
Well, goodness, as a complete outsider,
that's okay, but you know, just
ignorant of what is going on, I feel completely unequipped to be making recommendations. But I guess I will say one thing that from my experience in Uruguay and in working with researchers in Latin America, we are noticing a really big lack of biomonitoring and for metals, but also other chemicals. And that's one of the things that we're actually have a writing group together who's working on a call to recommend biomonitoring programs for individual countries as well as regionally to share data across the region, because it is really amazing in a bad way how the lack of data on exposures in populations makes for a very non-active policy arena. So I'll stop there, and again, take this with a grain of salt of someone who really shouldn't be giving any sort of recommendation in a setting that I've never been to.
Okay, thank you, Kasia. That's okay. Appreciate your input. Lynn?
Okay, maybe 3 points. The first one would be looking at the conceptual framework of the whole and pollution pathway from the source to pollution, that if we are going to work on a project, it has to touch all these 5 important components. And the second point would be is that I would still advocate a risk analysis thing, but taking into consideration the different nature of what we're dealing in right now, we have to shy away from the usual paradigm that we have about risk analysis, but still a good model, but something that we need to maybe revise and adopt when it comes to environmental pollutions, taking into consideration what I have said about toxic effects. And maybe the third thing is to be able to fill in the gap as far as that conceptual framework would be. I would still be advocating right now the biomonitoring and maybe harmonizing in terms of environmental monitoring, but most especially I think the point that Roy brought up earlier, we have to identify what is that biomarker that we want or that metabolite that we want that will have really significant effect in terms of health, because we can analyze so many, but we can prioritize in terms of what would really impact health at the moment. And the last would be is really identifying what will be the best methods, because this is now, as far as environmental health, we're challenged by what would be the best methods to be able to be, at the end of the day, establish that link from source to ill health. I think that's all. Thank you.
Thank you, Lynn. Michael?
So for me, of course, I'm a big advocate for biomonitoring, so that's what we've been doing. But I do hope that we do have this continuous, the real biomonitoring, where you're really monitoring with time. But I think that the challenge there is that we don't have a center where I think that maybe PAASE could advocate for developing or identifying a center. When I mean a center, it's not just a location, but a group of researchers that can form this group, and so that you can have an expertise.
Oh no, I think
Was Michael?
we lost Michael.
And that's Michael.
Oh, okay. Well, we'll come back to Michael. I guess the next one was Roy.
Yeah, okay, so that's timely because I'll just echo what Michael is saying. So based on the discussions that we've had, I feel like there are a few things that I think need to be addressed. One is turfing issue. So I would, of course, advocate for biomonitoring, but biomonitoring is such a huge undertaking. You have to really start small and show that that particular paradigm would have a success in terms of maybe changing the regulation or improving the health of Filipinos. So with turfing, I feel like, you know, if we assemble a group of scientists and other stakeholders, identify all the stakeholders, not just scientists, we need social scientists, communications expert, involved industry, because they're a huge stakeholder to this. I feel like, you know, it's almost counterintuitive when you talk about biomonitoring, you're involving industry. But I think, you know, when it comes to commercial viability of products sell because one, they're useful, and second, they're safe. And really we can approach that angle, improving the safety of the products in order to think of more of a sustainable viability for the product. So I would advocate, I guess, based on the discussions that we've had here, that we start with a small biomonitoring project consisting of all potential stakeholders and experts, not only scientists. Maybe we start with something familiar, so heavy metals, something that you're already familiar with, but also extend that to some environmental chemicals that already have a lot of baseline data across the world, so that when data comes in from the Philippines, one, we know that we have baseline data to compare it with, and therefore maybe use that comparison to further justify that there is really a need for biomonitoring in the Philippines because our levels are much higher than Indonesia or Japan or the United States. But really start small and ensure that maybe that small project would have success.
Thank you, Roy. Actually, also in the comments, like Dr. Katarzyna Banaay is saying, you know, similar things, like create a network of scientists working on environmental health. Mike, you were cut off. You were saying something about a center, a group of researchers that can provide expertise. So do you want to complete that thought?
Yeah. So I do want to highlight that we need to create a center because there's a disconnect between the different agencies. Now, for example, the DNR, whenever it comes to pollutants, you say they would say, the government would say, oh, it's DNR has to take care of it. No, it's DNR's forte. But I think because of that disconnect, they're not talking with the PCHRD, which is also the health side of the government in terms of funding. So if we have a center, then it's going to be easier to bridge this gap between the two different departments, from the environment to health, and then they will be talking about. And if we focus on the center, a center that focuses on biomonitoring or connecting pollutants, environmental pollutants to health, then that agency would also help lobby, help also maybe transform this to more the data into more laymanized terms so that people can understand. So it'll be good to have that interdisciplinary sort of a center that is focused on particular topic, in terms of pollutant. So that's pretty much what I wanted to say.
Thank you, Michael. And I apologize for extending over 10:30 for a little bit, but maybe just a minute or two, Alex and Lara. So go ahead, Alex.
Okay. Well, my original recommendation was to initiate a national program to develop superior metrics. But what I'm more afraid of is that it's just going to fall upon deaf ears in the government. And so I want to echo what Roy said and what Michael said, and especially listening to discussions. I think Dr. Lynn, I don't know her last name because I can't read the rest of the name, the physician, Dr. Lynn. I think she hit upon the main bottleneck, which is the fact that there are so many bureaucratic silos and scientific silos where people don't communicate with each other. So it's really hard to move things forward. And it makes me think about my background in the pharmaceutical and biotechnology industry for 30 years, where, and especially in cancer research, where drug development consisted of a series of silos where the biologists would find a drug target, hand it over to the chemist who would try to find a small chemical entity, but they would never talk to each other. And then the chemist would never talk to the toxicologist, and the toxicologist would never talk to the physicians who would conduct the clinical trials. And the success rate for developing cancer therapeutics was absolutely dismal. About 2.5% of all the ones that cured cancer in animals or in tissue culture, only 2.5% actually went on to phase 2 and 3 clinical trials. But then, about 20 years ago, everybody started to talk about translational research and translational medicine, where we break down the silos and have everybody talk to each other and have a common goal. The physicians talking to the basic researchers, using the molecular biomarkers. And now, the majority of the drugs that have been approved by the FDA are all these breakthrough cancer targeted therapies, like Levac, which had the fastest approval rate in the FDA history, and basically cures chronic biologic leukemia. We have targeted therapies for breast cancer. So I think, yeah, breaking down the silos could be one of the solutions through a mechanism like what Michael or Roy suggested. So maybe this group can maybe initiate the process towards that.
Thank you. Thank you. Sorry. Yeah. Thank you. And finally, Lara, if you can summarize in a few minutes. Yeah.
Yes. Before I go into that, I want to acknowledge Dr. Veronica Migo and Dr. Laurie Trinidad. They have been also part of our team for many years already. So for my part, I would say also that we would need to strengthen environmental health surveillance, as what Dr. Lynn already mentioned. And that would eventually lead us to really the need for biomonitoring, because it's a proactive approach. We don't have to wait for the worst before we act on something. And it also leads to informed decision making, where we know what we need to address, and we will know what to do where, and what segment of the population do we need to focus on. At the same time, I think that will help get the issues prioritized by the government and all other stakeholders. But I just want to note in there that we should be ready also to have a response system, because we cannot tell the people you are being poisoned and we cannot do anything about it. So yeah, that's it for me.
Thank you. So I again echo what Diana said earlier, which is I really appreciate the time of all the panelists and everybody who participated, who attended. Really appreciate your comments. There's really good comments also on the chat, and we're going to capture all of them. So, like Diana said earlier, this is also a venue for potential collaboration, research cooperation. Um, I think I think I can figure out your email from the registration on Zoom, and so maybe expect an email to everybody, so at least you'll see everyone's email if you want to connect with a specific person here, and so hopefully that will be the start of something. And then also for the panelists and other interested people, we're probably going to draft a very brief set of recommendations and then float it around. And so hopefully, you can give us your feedback so we can have at least something that we can start with. Appreciate everyone's time. Really, it was interesting and really enlightening. So I hope you found the topic and the discussion valuable. Diana, last word from you.
Oh, I don't have anything. Good night, everyone. Thank you so much.
Good morning.
Good morning. Good morning.
Good morning. Yeah.
Good night for us. So
Yeah.
thank you all. Thank you so much.
Okay.
You also.
Bye-bye.
Bye-bye. Thank you, everyone.
Bye.
Thank you, Roy.
Bye.